Let's start with red blood cells. Now, you cannot understand the process of erythropoiesis or the process which produces them without first understanding what are red blood cells, how they are composed, how they functions, and what makes them special. So, in order to learn that, first we have to look at the anatomy or the structure of the red blood cells. Now, red blood cells, as you can see in this diagram, they have a very typical biconcave disc-like shape to them. Now, what it means that they have a depression in a center while they bulge out towards the periphery. Now, this is a cross section of an one RBC. Here you can easily appreciate that in this cross section there is bulging out at the peripheral areas while there is thinning or depression at the central area. Now, this gives rise to the special or very particular shape that is only for the RBCs. Now, why do they have this particular shape? It is because they have to carry oxygen. And because oxygen is carried out or is taken from the RBCs by simple diffusion from the alveoli to the RBCs, they need to have maximum amount of surface area to them. So this shape provides them with maximum amount of surface area. Now, cross-sectionally viewed, you can see that at their maximum, that is on their periphery, they have the maximum length of about 2 micrometers, while at their minimum width, that is in the center, it is only about 1 micrometer. So they do not usually exceed the maximum limit of about 2.5 micrometers. Now, this is the cross-sectional area of a normal RBCs. Now, when an RBCs is viewed from the top or from the above, now here the diameter of a typical RBCs ranges from 7 to 7.5 micrometer. The maximum usually remains at about 7.5 micrometers. Now, when it is viewed from the top and when you actually study it, when you look at through the microscopes, RBCs are usually viewed from the top. So that's why you have to see or appreciate their appearance, because they will look like there's a bulging or concentration towards the periphery with a depression in between them. Next, now we know that there are a lot of RBCs which make up the blood, but their range lies from zero point 0.5 to 5.2 million per cubic millimeter. Now that's a lot of RBCs circulating in the blood. Now, if you take into account that there is about 5 liter of blood circulating at any time in a human being, so 0.5 4.5 to 5.2 million per cubic meter is a lot of RBCs. That's why they have a specific lifespan and they keep on recycling. And this is the reason we have optimum amount of oxygen being transported from the lungs to the peripheral tissue. Now, it is one because they have sheer number to them. There's a lot of number of them circulating in the blood. Second is because of their surface area. Now, another thing is that most of the RBC is empty. Half of it is empty. Only about 48% of total volume of any given RBC remains filled with cellular components. About 52% is empty. Now, because of this, half of the cell remains empty. It can take any shape. It can squeeze through small pores through the capillaries. It can reach areas which are not accessible by the other cells of the blood, and they do so via a particular process known as the diapedesis. Now, during diapedesis is a process where, because they have huge amount of empty space in them, they can squeeze through even the narrowest of capillary pores. And because of this, sometimes RBCs can take, if they're not properly matured enough, a bag-like shape. Next is that they contain only about 34% hemoglobin to them. That is, only 34 grams of hemoglobin is found in about 100 ml of blood or RBCs. Now, this is important because this is the metabolic limit of any given RBCs. Any RBCs cannot exceed this limit. It cannot exceed the quantity of RBC hemoglobin being produced more than 34%. It has to remain at that limit. An RBC cannot fill itself with hemoglobin. It will only remain at the maximum extent of 34%. And this is known as the metabolic limit of hemoglobin synthesis. Next is the oxygen carrying capacity of RBCs. Now, 20 ml of oxygen is carried about by 100 ml of RBCs or blood. So you can see that if 100 ml of blood carries oxygen, it carries oxygen in amounts of 20 ml. Now, in proportion to 100 ml of blood, this is a huge amount. So you can easily appreciate that the oxygen carrying capacity of RBCs is huge. 100 ml carries 20. So that reduces the number to about 20%. So 20% of blood at any given moment is carrying oxygen from the lungs to the peripheral tissues. Now, we have been talking about the oxygen carrying capability of the RBCs, and it is done from the lungs to the peripheral tissue. Now, in order to visualize that, we can do so by this diagram. Now, this diagram composes of, first of all, the RBC. This is the RBC with its cross section. This is the plasma, which obviously accompanies the RBC. This is the capillary wall, and the capillary wall contains both the plasma and the RBCs. Now, across this capillary wall in the lungs is present the alveoli. Now, these alveoli contains the inspired air. So they contain maximum amount of oxygen. So oxygen which is present in these alveoli has to be transported across the wall of the alveoli, then the wall of the capillary, then into the plasma, and then across the wall of the RBCs. Now, when this transport happens, it happens through simple diffusion. But these collection of membranes, the alveoli, the capillary membrane, and the RBC membrane, it is what is known as the respiratory membrane. So oxygen simply diffuses across the respiratory membrane from the alveoli to the RBCs. Now, once it enters the RBCs, it performs a couple of functions. Now, one of those is that it combines with hemoglobin. Now, hemoglobin that is deoxygenated is usually protonated, that is usually combined with another proton or a hydrogen atom. Now, when oxygen comes in contact with the protonated hemoglobin, it combines with the hemoglobin and releases the proton. Now, this proton, when released, has to be taken care of. Why? Because this can decrease the pH of inside the RBC and increase the acidity. And when pH is disturbed, the metabolic functions of the RBCs can be severely compromised. Now, because of this, they have to be taken care of. Now, they are taken care of via this method. What happens is that there is a certain process known as the chloride shift. Now, what happens in chloride shift is that chloride that is present inside the RBCs is exchanged with bicarbonates which are present outside the wall of the RBCs. Now, when these bicarbonates enter inside the RBCs, they combine with the proton And when they combine with the proton, what happens is they produced a very weak acid, and that acid is known as the carbonic acid. Now, the carbonic acid, even though is a weak acid, must be taken care of because again it will cause an increase in acidity. So, we'll come to the second most important part or the physiological function of the RBCs. The first one being that their transport and exchange of oxygen across the whole body. The second one is that they contain a certain enzyme, and that enzyme is known as the carbonic anhydrase. And carbonic anhydrase facilitates the reversible formation of the carbonic anhydrase into the water and carbon dioxide. Now, when that carbon dioxide is released by the carbonic anhydrase from the carbonic acid, it simply diffuses out along the respiratory membrane into the alveoli. From there, it is expirated out. So, an RBCs has two vital functions. One, it has to carry oxygen from the lungs to the whole of the body. Second, it has to neutralize the carbonic anhydrase, carbonic acid, and it does so by using the very specific enzyme known as the carbonic anhydrase. And when that happens, carbon dioxide that is liberated simply diffuses out and is exchanged with oxygen, and that carbon dioxide simply is expired.